depending on how they segregate, there's just stochastic variation in the inheritance pattern. So this patient, unfortunately, had received a very, very heavy burden of deleted mitochondria in her hematopoietic stem cell compartment. So we asked whether we could reprogram from these marrow fibroblasts, and indeed, once again, at low efficiency, Sumit was able to isolate iPS cells, which he could demonstrate carried the deletion. He could also quantify the degree of heteroplasm, so not only the number of cells, but the percentage of the deleted chromosome. And what he observed over time, which was quite fascinating, was a passage-dependent loss of the deleted mitochondrial genome. Now, by the time he had made this observation, he was already starting to do functional analysis of these cells, and he got to the point where he was able to subclone. And I should point out that because the retroviral integration for the reprogramming act as a unique genetic mark, so we know that these are all clonally related. This isn't a combination population where subclones are being spit out, but rather they all carry the same genomic integration. So by the time he got to subcloning, he was able to isolate, essentially completely purge what would be mitochondrial normal cell types. So what he was able to show by just cell growth, measuring colony area over time, was that these purged iPS cells would grow much more robustly than those that carried a significant burden of deleted mitochondria. And when he differentiated these into hematopoietic tissue, the tissue that was most markedly affected in this patient, what he saw, even with cells that had already only about 10% deleted mitochondria, that the purged ones had in fact more robust blood potential. So this suggested to us that simply culturing iPS cells affected a purging of this deleted genome, which was a very exciting prospect for us, because it creates patient-identical normal cell types. So as we were considering how we might turn these purged normal iPS cells into tissue that could be considered for transplantation for this patient's anemia, unfortunately, she succumbed to H1N1 flu in our ICU. But this is a general strategy for thinking about an approach to mitochondrial disease. So there are conditions that we have found difficult to model by iPS reprogramming, and one such condition is Fanconi's anemia. This is another one of the congenital bone marrow failure syndromes, which also has attendant with it a variety of other developmental defects, including skeletal abnormalities. It's well known as one of the hypersensitivity to DNA damaging agent syndromes, and this is related to defects in a core pathway that senses DNA damage, the Fanconi pathway. And it's diagnosed clinically when it's shown as a knockout. This is hypersensitivity by looking at chromosomal breakage. And what's fascinating is that there have been a number of knockout models in the mouse which don't mimic the development of human marrow failure. When you look very hard, you can see that there are hematopoietic stem cell defects, there are defects of number and competitive engraftment, but they don't appear to develop the same type of anemia. So for that reason, there's been a lot of interest in generating humanized models. And yet, when Juan Carlos Espicio of Belmonte's lab attempted to reprogram Fanconi tissue, been replicated independently by a number of these cells are markedly resistant to reprogramming. And what they published was that one had to perform gene repair in the fibroblast, affecting essentially a normal fibroblast phenotype, in order to generate the reprogrammed cells. There's since been reports in scientific meetings that some groups, using an enhanced efficiency of reprogramming, have been able to affect iPS Fanconi cells. But given the resistance, what Osman Tulpale and my group did was ask whether we could model Fanconi anemia now using shRNA-mediated gene knockdown. And so this is just an alternative strategy for using embryonic stem cells. Here he engineered into this short hairpin vector the knockdown of the FANK D2, here now quite effective at high multiplicities of infection, and the knockdown of the FANK A. And just quickly I'll say that by measures of protein, both immunofluorescence and Western, we affected a very nearly complete knockdown. And then he could show that the embryonic stem cells had the same sensitivity to DNA-damaging agents, and they generated these complex radial chromosomes so that they mimicked the cellular phenotype of the disease. And then by directing the differentiation of these embryonic stem cells into embryoid bodies and assessing their colony-forming activity to look at hematopoietic potential, what he could show by a number of assays was that they in fact showed a deficiency of hematopoiesis, both by CD45 positive hematopoietic quantification, the expression of known markers of the myeloid lineage, as well as defects in the ability to form myeloid colony-forming cells. So here's an independent strategy where we've had trouble modeling the disease using iPS cells, but we've been able to make insights into very early hematopoietic defects using the knockdown of the Fanconi anemia gene in human embryonic stem cells. And then my last example is one where, once again, we see a different and perhaps advantageous modeling of a human disease using embryonic stem cells. And that condition is Fragile X syndrome. And this is a well-known, very common form of genetic mental retardation that involves the silencing of a gene that carries a triplet repeat in its 5' untranslated region. And Nassim Ben-Venisi's group, indeed a graduate student, Ahia Urbach, who worked with Nassim, generated an embryonic stem cell from Fragile X embryos that were diagnosed through pre-implantation genetic diagnosis. And what they showed was that this FMR1 gene is expressed in the embryonic stem cell, but undergoes differentiation-dependent silencing, so that fibroblasts that are derived from in vitro differentiation have now undergone active gene silencing. This mimics what's been observed in human development, where samples of chorionic villus tissue of the early developing Fragile X individual express the FMR1 gene, but by the time the individual has matured, there's active silencing of the FMR1 gene, and there's fabulous new work suggesting the relevance of this FMRP in the neural synapse act as a regulator of RNA expression, and in particular, perhaps a tonic decrease in the expression of the metabotropic glutamate receptor, which is very exciting because there are drugs that antagonize this receptor, and these are now in clinical trials. So Ahia, working on this as a graduate student, then came to my lab as a postdoc and asked first whether or not one could use iPS cells to mimic this condition. So what he asked first was, what is the expression of the FMR gene in both here the control embryonic stem cells, but wild-type fibroblasts and iPS cells, and it's indeed all expressed. But in fact, it's not expressed as expected from 3 different patient fibroblasts. And then the question is this locus reactivated by the reprogramming process, the way the entire rest of the genome is reset? And indeed, across about a dozen different clones from 3